Recording, share the screen. All right, can you see my screen?
Yeah.
All right, thank you. Let's close that. So if you check the schedule, we're going to be continuing Chapter 8, Microbial Genetics today. In the lab, we are not meeting today, so I will be there from 6:30 to 6:45 to answer any questions. At 6:45, if there are no questions, I'll then log off. Remember, your infectious disease research project is due this Saturday, February 20th at 11:59 p.m. Also work on your unknown work, and if you haven't done Lab 8, please finish Lab 8. Any questions? I am a little bit behind on grading the labs. I apologize for that. I think I just graded Lab 6, if I remember correctly, earlier this week. And I'll try and get to Lab 7. I'll try and get to Lab 7 at least by this weekend. Any questions about the quiz you just took yesterday? All right, let's move on. So we were talking about genetics, and we finished talking about a complication where in prokaryotes, when the messenger RNA is being transcribed, translation can start on the messenger RNA before transcription finishes. And there can be many ribosomes attached to that one messenger RNA, which is not finished being transcribed yet. Everyone clear on that? The reason why that happens in prokaryotes is the DNA is in the cytoplasm, so the messenger RNA gets made in the cytoplasm, and then the ribosomes are in the cytoplasm and they can attach to the messenger RNA, and that's why it all happens at one time. Any questions about that? Go ahead and delete that. Well, maybe that got rid of it. Yep. There is another complication in transcription that's only a complication in eukaryotes, and that is the RNA in eukaryotes has to be processed before it can leave the nucleus. And why it has to be processed in eukaryotes, a gene right there contains exons and introns. Exons are the portion of the DNA that actually codes for the gene products, meaning if the gene codes for a protein, the exon is the part of the gene that codes for the protein. The introns do not code for the product. They do not code for the protein. And eukaryotes have genes that are composed of exons and introns, and the introns have to be spliced out of the RNA to get the final messenger RNA. So when transcription occurs, all of the DNA is transcribed into RNA, and the introns are spliced out. That means cut out. And then the exons are spliced together. That means joined together. and then that will be the final messenger RNA, and only the final messenger RNA can leave the nucleus, meaning if the RNA has introns in it, I'm not sure how that works, but the introns are a signal to the pore in the nuclear membrane that RNA transcript cannot go through the nuclear membrane. So not until the introns are removed can the final messenger RNA leave the nucleus. Any questions about any of that? Okay, when the final messenger RNA, which is only exons, the introns have been removed, leaves the nucleus, it can then go into the cytoplasm and be translated by the ribosomes. And remember, ribosomes are in the cytoplasm. Sorry, I need to move this a little. All right, so we talked about the structure and function of the genetic material. Now let's move on to regulation of gene expression. And we're going to focus on the regulation of bacterial gene expression. Eukaryote gene expression is similar. It does use operons to control gene expression. It's just that the control is more complex and there's more regulation of gene expression in eukaryotes. And then after talking about the regulation of bacterial gene expression, you'll learn about mutation. That should be in the worksheet, the in-class activity for this lesson. And I think you have 2 in-class activities in Chapter 8. And then we'll talk about the genetic transfer and recombination. And then lastly, we'll talk about genes and evolution. Any question about any of that? I stood up to get out of the camera because I had to blow my nose. That's not a cold and it's not COVID-19. It's just the weather is changing and it makes my nose run. Regulation of bacterial gene expression. One way to regulate a cell is to control gene expression. There are other ways to regulate gene expression or to control a cell, and we did talk about a few of them, and that is you can have feedback inhibition, turning off an enzyme, and that's regulating the cell by not allowing the enzyme to function. You can regulate it also by turning on and off the genes. Constitutive enzymes are enzymes that are expressed at a fixed rate. Some genes are always turned on, and they're turned on at a fixed rate. There are other genes that are only expressed when they are needed by the cell. And these genes we call repressible or inducible genes. And we'll talk about them with the repressible and inducible enzymes. The regulation of these genes occurs by pre-transcriptional control, meaning you are controlling gene expression before transcription. And this happens in the operon, and we're going to talk about it with inducible operons. There are repressible operons, and I think your book talks about it, but you're not going to be tested on it, and I'm not going to cover it. There's also epigenetic control, and you're not going to be tested on it, and I'm not going to talk about it. And epigenetics is really something that even geneticists don't really understand, and they try to explain it, and they wave their arms, and it's complicated. The gene is controlled by non-genetic means, okay, epigenetic control. And then there's post-transcriptional control, which we're not going to be tested on it, and I'm not going to talk about it further, although, like I said, whenever you inactivate an enzyme, that is post-transcriptional control. There's also post-translational control, like you can take the enzyme and degrade it. But we're not going to talk about post-transcriptional control from here on in this lesson. We're only going to talk about operons, and specifically inducible operons. Now, all genes are not turned on all the time. And why? It's because it doesn't make sense for the cell to turn on a gene if it's not going to use the product because it's not needed. Does that make sense?
Yes.
So if the cell doesn't need an enzyme, why make it? It would be more efficient not to make it if you're not going to use the enzyme. And that is regulated by an operon, frequently by inducible operons, and we'll talk about an inducible operon. What a bacterial operon is a model for control of gene expression, and what an operon is the gene of interest, which is given the name a structural gene. And usually when we talk about a gene of interest, we are talking about a structural gene. For example, the enzyme lactase is a structural gene, meaning there's a gene that codes for the enzyme lactase. When we talk about the blue-eye gene or the brown-eye gene in people, we're talking about a structural gene. An operon includes the structural gene, and there may be more than one structural gene in that operon, and then the controlling regions of the DNA around the structural gene. So you can think of the operon as the super gene. It includes the gene of interest and then the controlling regions around that gene, and it's not labeled here, but the terminator that ends the gene. In prokaryotes, you can have more than one structural gene in an operon, and these other structural genes are all involved in the same pathway. Like for the lac operon, the Z gene codes the enzyme lactase to break the sugar lactose down into beta galactose and glucose. The Y gene is the transport protein that brings the sugar lactose across the cell membrane. And the A gene is the gene that is helping to convert the sugar beta galactose into glucose. All cells can metabolize glucose, but the other sugars have to be converted into glucose to be metabolized, and the A gene is one of the genes that's helpful in converting galactose into the sugar glucose. And so all of the structural genes are involved in metabolizing the sugar lactose. Any question about the structural genes? Eukaryotic operons are similar to prokaryotic operons, but there is one exception. Each operon in eukaryotes has one and only one structural gene in the operon. And we humans have the Z gene that encodes the enzyme lactase, and the Y gene that allows the sugar lactose to get across the cell membrane, and the A gene that helps convert the sugar galactose to glucose. But we have them in 3 operons. In prokaryotes, they have it in 1 operon. Any questions about that? So in one sense, eukaryotic operons are simpler because they always have one and only one structural gene in the operon. But in another sense, they are more complex because they have much more regulation of the operon. And we'll talk about regulation in just a minute. One way to regulate the expression of an operon, meaning the structural genes in the operon, is to have a regulatory gene, and that's shown in this map. The regulatory gene is not part of the lac operon. So only this part is the operon for the lactose metabolism pathway, and we call that the lac operon. This gene makes a product that regulates the expression of the operon. And in fact, this gene here has its own operon, and we're not going to look at that. We're just going to look at the lac operon. Any question about that? All right, the P we've already talked about. That's the promoter site. That is a regulatory region that what it does is on the DNA, the promoter codes for the RNA polymerase to bind to the DNA. And then the RNA polymerase transcribes the DNA to make the RNA. And so from here on down, RNA is transcribed. And the terminator codes for, which is at the end of the gene, it's not shown here, codes for the RNA polymerase to fall off the DNA. And all of that is part of the operon. Any questions about the operon? The lac operon, which means the lactose operon, is an inducible gene. That means it is normally turned off and is only turned on when the cell needs the enzymes to metabolize the sugar lactose. Any question about any of that? Okay, let's talk about the regulation of the lac operon. Remember, regulation can happen by induction. That means the gene is normally turned off, but it can be turned on when the products of the gene or the lac operon are needed. There's also repression. That means the gene is normally turned on, but it can be turned off by regulation. And we're not going to talk about a repressible operon. I think your textbook talks about repressible operons, and you can read about it if you want. The lac operon is inducible operon. What happens is the repressor gene, which is this I gene here, which normally is not near the gene or the operon it controls. But in the case of the lac operon, the regulatory gene, the I gene, is right next door to the operon. And in fact, when we're talking about eukaryote operons, the regulatory gene frequently is on another chromosome. In fact, usually it's on another chromosome. And then it makes a product which regulates the operon it controls. All right, any question about regulation by the regulatory gene?
No.
I'm not hearing anything, so I'm going to move on. The regulatory gene in the lac operon is transcribed, and it makes a repressor protein by translation of the messenger RNA. And that repressor protein will bind to the operon at the site of the operator. So let me back up. The operator is the second controlling region in the operon. Okay? And the regulatory gene makes a repressor protein that binds to the operator. Now, when the RNA polymerase binds to the promoter and then moves down the DNA, it runs into the repressor protein bound to the operator. And then the RNA polymerase cannot move further down the DNA. And that will shut off gene expression for the genes in the structural genes in the operon. Meaning, if we're talking about the Z gene, no messenger RNA for the enzyme lactase will be made, so no protein will be translated, and there will be no enzyme lactase in the cell. Okay? Any questions about that? So that is how in prokaryotes the lac operon is shut down, and is shut down most of the time. The I gene is the repressor gene, and it makes a repressor protein which binds to the operator, and that stops the RNA polymerase from moving further. And then no enzyme lactase, as well as the other proteins, will be made. In E. coli, why would E. coli normally shut down the lactase gene? This is a question. Is anyone going to answer? Why do you think in E. coli it normally shuts down the lactase gene?
So it can grow?
Well, yes. But what does the lactase gene do? Come on, somebody's got to know. What does lactase do?
Fermented?
It what?
It does fermentation, no?
It breaks down the sugar lactose, if that's what you mean. Okay? And why would E. coli not want to do that? Why would E. coli not want to break down the sugar lactose? How often does E. coli see the sugar lactose? Okay, I'll ask this. Where do you find the sugar lactose?
Would E. coli rather use glucose as an energy source?
It does, but I'm not asking that question. I'm asking how often would E. coli see the sugar lactose? Where do you find the sugar lactose? Come on, somebody's got to know. Where do you find the sugar lactose?
Lactic acid. But in dairy products?
Yeah, in dairy products. How often is E. coli going to be around dairy products?
Very often.
Why do you say that?
Well, that if it's in normal circumstances, I mean, it's normally present in a lot of lactose, in fermented foods, so.
Yeah, E. coli might be in fermented foods, but it's not a bacteria we use to ferment those foods. If you look at any of the dairy products, they don't add E. coli to make yogurt or to make cheese.
isn't E. coli normally found in like chicken?
You find E. coli actually all over the place. You do find it in the gut, but like for me, I'm lactose intolerant, so I don't eat dairy products, or at least not much of them. So E. coli in my gut is not going to see the sugar lactose. And if the E. coli is on a apple or in the soil, it's not likely to see the sugar lactose, because lactose is not a very common sugar. You see it in dairy products, and that's it. So if the E. coli happens to be on a dairy farm, and the E. coli happens to fall in the bucket of milk that the cow farmer is milking the cow out of, the E. coli will actually see the sugar lactose. But at most other times, E. coli is not going to see the sugar lactose, because lactose is not that common. You only find it in dairy products. So, E. coli normally turns off the lac operon, because E. coli is normally not going to see the sugar lactose. It's not a common sugar. E. coli is
You're
much
right.
likely.
You're right, because you know, most dairy products in this country are ultra pasteurized. So yeah, we'll have like very small chance to activate like that.
That's true, but like I said, in dairy products, we don't use lactase or E. coli to make the dairy product. Take a look. I'm trying to remember some of the names. Acidophilus is one of the bacteria we use to ferment yogurt. I don't remember the other species, but like I said, we don't use E. coli to make dairy products. We use other bacteria. Well, here's another one. Is it lactobacillus or lactococcus? One of those two is used to make dairy products. So E. coli makes the repressor protein that then binds to the operator and shuts down the lac operon, because E. coli normally is not going to see the sugar lactose. But if lactose, the sugar lactose, is around, the sugar lactose can get into the E. coli, and whenever the sugar lactose is around, the stereoisomer, which is the right hand of the left hand, meaning one is the same molecule, it's just one is left-handed and one is right-handed, if you remember that. Allolactose is the stereoisomer of lactose. And if lactose is around, you always have allolactose around. Did that make sense?
Yes.
Okay. And allolactose is the inducer. Allolactose will bind to the repressor protein and inactivate the repressor protein. If all of the inactivated repressor protein is inactivated, then none of it can bind to the operator. And then when the RNA polymerase binds to the promoter, it can move down the operator and then transcribe the lactase enzyme and the other genes of the operon, and then make the enzyme for lactase. Beta galactosidase is the same as lactase. Lactase is just a shorter name. Okay? So how the operon gets turned on is whenever lactose is in the cell, there will also be allolactose. Allolactose will bind to the repressor protein and inactivate the repressor protein. And that will turn on the gene expression. Okay?
So would that turn on only if glucose wasn't there and it needed lactose or lactase, excuse me?
No. If glucose is in the cell and lactose is in the cell, which actually, whenever lactose is around, it will be broken down into galactose and glucose. So whenever lactose is around and the lactase enzyme is around, you'll have the sugar lactose, lactose and glucose in the cell. Does that make sense?
Yes, thank you.
Okay. So I've got a question for you, and that is the allolactose, the inducer, inactivates the free repressor protein, but it doesn't bind to the repressor protein if it's bound to the operator. So how do we have the repressor protein that's bound to the operator fall off of the operator so the inducer can bind to the repressor protein? Do you want me to say that again?
Yeah, can you repeat that?
The inducer binds to the free repressor protein, not to the repressor protein that's bound to the operator. So to get this gene or the operon turned on, we have to have the repressor protein not on the operator. My question is, what happens to the repressor protein that is initially on the operator? The inducer can't bind to it here. So how do we go from here to there to turn on the lac operon?
Does the inactive repressor protein just act as like a messenger to the, that makes sense?
I know what you're saying. You're saying that the inactivated repressor protein makes the repressor protein fall off of the operator. Not directly, but you are right in a sense, and that is, if it's all inactivated, that's going to encourage the repressor protein to fall off the operon. What happens is the repressor protein binding to the operator, that's not a permanent bond. It's a stoichiomic reaction, and every once in a while, the repressor protein, when it's bound to the operator, it will fall off. Okay? So, if the repressor protein is inactivated by allolactose, then sooner or later the repressor protein bound to the operator will fall off because that binding is a stoichiomic reaction. It will fall off eventually. And then when it falls off, it's now free to bind to the allolactose, and there's lots of allolactose around, and it will inactivate that repressor protein so it cannot rebind to the operator. And that will turn on the gene expression for the lac operon. Any questions about that? So whenever lactose is around, we have allolactose around, and it will inactivate the repressor protein, and then the repressor protein that's bound to the operator will sooner or later fall off and become activated, or not inactivated, by the allolactose. And that will turn on gene expression, and then we will transcribe and then translate the structural genes in the operon. And then let me ask another question here. What happens to this RNA polymerase that's stuck on the promoter operator region because the repressor protein is bound to the operator? Does it just stay there forever? That's a yes or no question, people. You have a 50% chance of getting it right.
No.
That's correct. It does not stay there forever. It binds in another stoichiomic reaction, and every once in a while the RNA polymerase will fall off, and then it'll bind to some other promoter. So it's not permanently stuck in this position. Any question about any of that? Okay, no question about that. Let's watch a little video on the lac operon.
The E. coli lac operon is an example of an inducible set of genes. These genes are responsible for the breakdown of lactose into sugars used for cellular metabolism. This inducible system also involves bacterial DNA, a repressor, mRNA, and the sugar molecule lactose. This animation will only focus on 2 of the 3 proteins encoded by the lac operon, beta-galactosidase and permease. Gene expression can be induced or turned on when a specific inducer molecule appears in a cell. For inducible systems, a repressor molecule prevents gene expression by binding to the upstream controlling region. Lactose is the lac operon inducer molecule. After first appearing in the cellular environment, lactose passively enters the E. coli cell and binds to the repressor molecule. This binding releases the repressor from the controlling region.
Okay, let me state that that is not quite true in the video, and that is the lactose does not bind to the repressor protein when the repressor protein is bound to the operator. They got that a little wrong in the video.
And binds to the repressor molecule. This binding releases the repressor from the controlling region. At this point, RNA polymerase can begin transcription of the operon. Here we show 2 of the 3 lac operon genes being transcribed into mRNA. Ribosomes then bind to the mRNA, and the 2 proteins are translated. The first protein is beta-galactosidase, which breaks down lactose into 2 simple sugars. The second protein is permease, a membrane-bound protein. When embedded in the cell membrane, permease functions to provide a direct route for the lactose outside the cell to be imported into the cell. This import occurs at a much greater rate than the passive transfer we first observed. Because translation continues inside the cell, other permease proteins become embedded in the membrane. This further increases the rate at which lactose enters the cell. Beta-galactosidase breaks the cellular lactose into the simple sugars glucose and galactose. Once its concentration is greatly reduced, the lactose bound to the repressor are released. At this point, the repressor again binds to the controlling region and gene expression is halted. For all inducible systems like the lac operon, it is the interaction of the repressor and inducer molecules that mediate gene expression.
All right, any questions? All right, if there's no questions, let's move on. Anyways, that's the lac operon. Let's talk briefly about mutation. Mutation is a change in the genetic material. Mutations may be neutral, meaning they do not benefit an organism. They could be beneficial, or they could be harmful. Most mutations are either harmful or neutral. Mutagen is an agent that causes mutations. Like if you don't know, cigarette smoke is a mutagen. What's another mutagen? Benzene is a mutagen. Spontaneous mutations do occur, and whenever the DNA is replicated, mistakes can be made, and that gives rise to spontaneous mutations. And they occur in the absence of a mutagen. Here's a mutation in somebody's eye compared to a normal eye. There are different types of mutations, and I'm not going to discuss them because it's in worksheet lecture 8B. But you need to know the different types of mutations. So let's go to the end of that worksheet. And the worksheet also talks about the different types of mutations, how you can have spontaneous mutations and then mutations caused by a mutagen. And there's two types of mutagens, radiation and chemical mutagens. All right, I think this is where it ends. I don't have a statement here. Let me just check to make sure. Yeah, I'm pretty sure that's where it ends. Oh, there it is. I just didn't see it. All right. So when we're talking about mutation, there is a rate that spontaneous mutations occur at. And I don't know if this is in humans or E. coli, because they say one rate here, and it could be that it's very similar between most organisms, and bacteria and humans might have the same spontaneous mutation rate. But in general, the spontaneous mutation rate is one mutation in 1 times 10 to the 9th replicated bases, meaning when the DNA polymerase is copying bases, it'll make a mistake 1 in 10 to the 9th replicated bases. Any question about that? This is meaning that a mutation happens once in 10 to the 6th replicated genes, or once in a million genes. And the reason for that is that most genes are about 1,000 nucleotides long, and so the mutation rate per gene is 1 in 10 to the 6th. So if you look at humans, because we're over 1 million in population, in fact we're over 1 billion in population, you will always have a mutation for whatever gene you look at. Like if we look at the blue-eyed genes, you only need to look at 1 million people, and then you will find one person who has a mutation in their blue-eyed gene. And it's a good question. What does that mutation do? I don't know. I can tell you that about 10,000 years ago, there was only brown eyes in the human population, and then about 10,000 years ago, there was a mutation in the brown-eyed gene in Northern Europe, creating the blue-eyed gene. And we'll call it an allele from here on, because I'm talking about two different alleles now, the blue-eyed and the brown-eyed gene. And for some reason, in Northern Europe, that blue-eyed allele increased in frequency, so that now in Northern Europe, the blue-eyed allele is now the dominant, meaning the predominant allele. It's found more often than the brown-eyed allele in Northern Europe. When we look today, we cannot find any selective advantage for the blue-eyed allele over the brown-eyed allele. But they think a long time ago, meaning 10,000 years ago, they think the blue-eyed allele had a selective advantage. And that's why it increased in frequency. Any questions about any of that? All right. Mutagens increase the mutation rate. You may increase the mutation rate 10 times. You may increase it by 1,000 times. So it increases the mutation rate instead of 1 in 10 to the 6 replicated genes, but 1 in 10 to the 3 to 1 in 10 to the 5th replicated genes. Any question about that? So if you're a cigarette smoker and you're planning on having children, you will increase the risk that your child will be mutated if you continue to smoke. That's just a thought to think about. Any questions about that? Are these numbers explanatory? Do you guys follow this? There's 1,000 bases in a gene, so that means that if this is the base mutation rate, the gene mutation rate is 1 in 10 to the 6. And if you increase the mutation rate tenfold, you will make the mutation rate 1 in 10 to the 5, which is right there. All right. Identifying mutants experimentally. We can test for a mutant. You can test for the production of mutants or just the detection of mutants. You can do it with a positive direct selection that will detect the mutant cells because they grow or appear different. Like if we were to see somebody walking down the street like this, you'd say, yep, that's a mutant, right? No comment. That's supposed to be a little bit funny.
It was good.
Anyways, you can see that that's different. A negative or indirect selection is detecting the mutant cells because they do not grow. And so we're looking at the altered phenotype because they do not grow, and I'll explain that in the next slide. It's more difficult finding these, but it's not impossible. I actually did this. What you can do is you get a plate that has isolated colonies on it, and how you get that is you just take a solution of bacteria and you dilute the solution so that you'll get about this many colonies on the plate, and then you let it grow up and there will be isolated colonies like that. And what you do is you get a velvet surface and put it around a handle and then stamp the velvet onto this plate of bacteria. And then you stamp the velvet onto a clean, fresh, new plate that is the same media containing the amino acid histidine in it. And then you stamp the velvet onto another plate that is another media, and this media lacks the amino acid histidine. So the medias are the same, it's just that this one lacks the amino acid histidine. And you see this bacteria cells here do not grow on the plate lacking the amino acid histidine, but they do grow if you supply the amino acid histidine. So that these cells are mutant, they will not grow unless you supply the amino acid histidine. And this is one way to check for negative selection of a mutant. And when I was in college, I actually did this, stamping plates, and we were looking for mutations. Any questions about that?
What kind of description will you see? What kind of I mean, how can you spot a mutation in a plate like that?
Well, you see, this colony didn't grow, and this one did. It came from
Okay.
the same stamp. These cells here. So the cell that formed this colony was a mutant, and it has to be supplied the amino acid histidine, because to this cell, amino acid histidine is an essential amino acid, and it will not grow unless you supply that amino acid.
Interesting.
These cells here will grow if you do not supply the amino acid histidine, so that the cells can make the amino acid themselves from some product. Usually they'll make it from another amino acid, but I don't know. I'm not a biochemist. I can't tell you how you make the amino acid histidine. But these cells can make the amino acid histidine, okay?
I see.
But these cells can't. So they're a mutant. And that's negative selection. All right, any question about mutations? All right, let's move on to genetic recombination. Recombination is the exchange of genes between two DNA molecules. So on a chromosome, like the X chromosome from the mother and the X chromosome from the father, you can get recombination of those two chromosomes. And that happens with crossing over. Crossing over is literally the crossing of two chromosomes and then the breaking of those chromosomes and then the reannealing, so that if this is the mother's chromosome, it's all lowercase alleles, and the father's chromosome, the blue one, all uppercase alleles. The chromosome can cross over and then break off, and now the mother's allele is lowercase e, lowercase f, lowercase g, but then uppercase H and uppercase I from the father's chromosome. And that's a crossing over. When two molecules cross, break, and then rejoin. Okay? Recombination is the exchange of genes between two DNA molecules, which a crossing over is a recombination. Crossing over is just a little more specific, talking about the crossing of two chromosomes and then they're breaking and then reannealing. Recombination is referring to genes, so it's a little more general, but they are a similar term. Okay, recombination is a little broader. Like you could have the gene come in from outside the cell, and it might only replace the I, and we'll call it a double I yellow color because it's not from the father. And that would be a recombination event, not a crossing over event, because the gene came from outside the cell. Okay, so do you understand what crossing over is and what recombination is? And most of the time they're the same, but recombination is a little more general, and it does include a little more than crossing over.
When you say most of the time they're the same, when would they not be the same?
I just gave an example. If a gene were to come from outside the cell and then were to recombine with, let's say, let's see, I said the mother's was lowercase. You know, they changed the color here and there. If that gene were to join to the father's chromosome and replace the capital I with a double capital I, and it's going to be a yellow color now because it's a gene from outside the cell, that's not a crossing over event. but it is a recombination event.
Okay, I get it now. Thank you.
Okay? So recombination is a little more general and covers a broader topic than crossing over. Most of the time, though, they are the same. Like in our cells, when we have recombination happening, like between two X chromosomes in women and girls, that would be a crossing over event. Of course, men won't do that. The Y chromosome doesn't cross over with the X chromosome. But we could have in men the mother's first chromosome can cross over with the father's first chromosome. Okay? All right. Let's move on to genetic transfer. Genetic transfer can happen in 3 different ways. And I think I'm talking about it can happen in bacteria in 3 different ways. Conjugation doesn't really happen in humans. Transformation doesn't really happen in humans, although you could theoretically have that happen in humans. But all 3 of these can happen in bacteria. Transduction can happen in both bacteria and humans. Genetic transfer doesn't usually happen in vertical gene transfer. That's occurring during reproduction or between generations of cells. So the parent cell gives rise to 2 daughter cells, or 2 parents in humans give rise to 1 offspring. That's vertical gene transfer, where the DNA is passed from 1 generation to the next generation. Genetic transfer usually happens with horizontal gene transfer. That's the transfer of genes between cells of the same generation, or between a donor cell, the cell giving up the DNA, and a recipient cell, the cell taking up the DNA.
David,
Let me mute
Javier,
that person. Just a minute, I'm having trouble here. Somebody's got their... I'm muting you, Denise, because we were getting sounds. Now let me go back to the share screen. See, that's a little complex. I had to do that to do that. Horizontal gene transfer can happen in 3 different ways: in transformation, conjugation, and transduction. Let's talk about transformation first. Transformation is where 1 cell gives up its DNA, and then we'll have naked DNA out in the environment, and a recipient cell picks up that naked DNA, brings it into the cell, and then some of the genes of that DNA can recombine with the chromosome of the cell. And in this case, we take up the A gene, and then that A gene can be expressed in this recipient cell. That will make this cell a transformed cell because it is expressing a new gene, the lowercase a gene, which was in the DNA the cell took up. The cell had the uppercase A gene. Okay? And we're not going to go into this, but that DNA coming into the cell generally has to recombine with the host chromosome, and it usually will recombine in a region of the DNA that's similar to that gene that we're bringing in. We're not going to go into that, but the idea is this cell takes up DNA, foreign DNA, and then can express some of that DNA, the lowercase a gene, and then we can say it is a transformed cell. It's a transformed cell because it's expressing a new gene and it's making a new gene product, the lowercase a gene. And here's a little picture of that. The recipient cell takes up some naked DNA, gets a little indigestion from the naked DNA, and then becomes transformed as it expresses the genes on the DNA that it took up. Any questions? Transformation is called the process of taking up naked DNA into the cell, but it is also, you can say that the cell becomes transformed, changing its phenotype from what it was to what it will become. Okay? So transformation can be used in two ways. Any question about that? And I'm not going to get into it, but whenever the cell changes its phenotype, we say it is transformed. Okay? Any questions about any of that? That is the first way that genetic transfer can happen. And this does happen in bacteria. I wouldn't say all the time, but it does happen in bacteria. And let's say that the gene that it takes up encodes for antibiotic resistance in a cell that is sensitive to that antibiotic. This cell will now be antibiotic resistant. And that cell could get antibiotic resistance by mutation, but mutation happens, let me write it here, mutation happens at 1 times 10 to the 6th. And I'm going to have a hard time doing that exponential. I don't know if I can do that here. Oop, didn't get it. Yeah, I can't do it here. So 1 times 10 to the 6th, meaning mutation happens in a gene only once in a million genes, or once in a million cells. Okay? And how often do you think transformation of this antibiotic resistance happens? Anyone want to guess? Does it happen more often or less often than spontaneous mutation?
Less often.
Less often. That's a good guess, but you want to guess again? You don't want to guess again. The other answer is more often. Yeah, it happens about 10 times more often. Once in 10 to the 5th cells, we have transformation occurring in bacteria cells. So if you're in the hospital and you get an infection which is antibiotic resistance, it's more likely to be from transformation than it is to be from spontaneous mutation. That's just some things to think about. All right, let's move forward. Conjugation is when one cell gives some DNA from that cell to another cell. And that can happen in bacteria in two ways, by sexual conjugation. In gram-negatives, sexual conjugation happens with the sex pilus. The sex pilus is this structure coming out of this cell here. And the sex pilus literally skewers an F- cell. This is the F+ cell. We'll talk a little bit more about what makes a cell F+ and what makes a cell F-. The sex pilus can actually literally skewer this cell and then act as a winch to pull the two cells closer together. They don't have to be touching, as shown in this this picture where they're almost touching? Because the sex pilus is hollow, and then a plasmid, which is the F plasmid from this cell, can copy itself and then send one copy into the F minus cell. And that's how gram-negative cells engage in sexual conjugation with sex pilus. Okay? Conjugation can happen in gram-positives. We're not going to talk about it very much, but what happens is the two gram-positives actually touch, and when they're touching, the cell membranes between this cell and that cell touch and then fuse so that there's no cell membrane there. Okay, so the cell membranes are like this, they touch, and then they fuse together, and then they open up. Okay, so there's actual exchange of cytoplasm between this cell and that cell. And that can allow DNA, usually a plasmid, from one cell to move into the other cell. All right? And don't ask me what happens to the cell wall. I don't know. I'm not an expert on conjugation and gram-positives, but this, when it happens, we call that a mating bridge in gram-positives, where the cytoplasm and the ingredients in the cytoplasm can move from one cell to another. Okay? All right, any question about any of that? If not, let's look at conjugation in a little more detail in gram-negatives. The sex pilus from this cell skewers this cell and then brings the two cells closer together. The sex pilus is hollow, and this cell has the F plasmid in it. It's called the F factor. The F plasmid can replicate itself and then send one copy of the F plasmid from this cell into that cell. And there we're seeing it moving from this cell into that cell. And now both cells have the F plasmid in them. And then this cell, which was F minus, is transformed into an F plus cell. An F plus cell has the F plasmid in it, and the F plasmid codes for the sex pilus, so this cell will grow a sex pilus, and these two cells will now separate. Okay? And then this formerly F minus cell, that's now an F plus cell, once it grows the sex pilus, it can conjugate with the F minus cell. So this newly F plus cell can now act like this F plus cell and then conjugate with an F minus cell. Any question about sexual conjugation? Once again, sexual conjugation can happen at 1 times 10 to the 5th times, or about 10 times more common than spontaneous mutation. Any questions about that? And here's a joke for you. Hopefully you'll get it. If you don't know why conjugation is sometimes called sexual conjugation, ask your significant other. Okay? No comments. All right. The third way that genetic transfer can happen is by transduction, and this is a little bit complex because we haven't talked about viruses yet, so let me blow this up. Transduction occurs between a donor cell donating DNA and a recipient cell using a virus. Normally, when a bacteria virus infects a cell, it will bind to the bacteria cell and then send its DNA into the cell, and then this DNA and the bacteria DNA will destroy the bacterial DNA because this bacteria DNA, the viral DNA wants to run the show in the cell. It wants to control the cell. And what it's going to do is use this cell to replicate more viruses and to control the cell. An easy way to gain control of the cell is to destroy the bacterial chromosome. So one of the first things it does is it cuts the bacterial chromosome. And then the bacterial DNA makes copies of itself, because that's what viruses do. They like to replicate themselves. And I gotta go to the next slide here. Nope. Why is this not being shown all? Anyways, the replicated DNA will then be transcribed, and then viral proteins will be made. And then the virus will assemble, and then it'll lyse the cell to release the viruses. And then that's how bacterial viruses replicate themselves and make copies of themselves and lyse the cell. Okay? Any questions about that part? Transduction occurs when a mistake gets made. Viruses make so many copies of themselves, they're not worried if a mistake is made. And as you can see, the viral DNA is reddish. The bacterial DNA that's cut up is bluish or purplish. And when the viruses are assembling, they bring in DNA into the virus. And every once in a while they make a mistake, like for that virus and that virus. Instead of viral DNA going in the virus, bacterial DNA goes in the virus. And viruses aren't worried because they make lots of copies to themselves. They aren't worried if they make a mistake. Their job is just to replicate and then make more viruses. And if they make a mistake, well, that's just too bad. They just want to make more viruses. And mistakes are okay. Anyways, if we follow what happens to this virus, it goes on and does the same thing, making more viruses. But if we follow what happens to this virus that has the DNA from the bacteria cell in it, that virus will infect another bacteria cell, and it will inject the DNA that's inside that virus into the cell. But note that this DNA is not viral DNA. So it doesn't code for the destruction of the chromosome of this bacteria cell. And then the DNA that it brings to this cell is DNA from this cell. And so this cell can incorporate that DNA inside itself and then express the DNA that it got from this cell. And this is transduction, where this cell becomes a recombinant cell and expresses DNA that it got from this cell. Okay? And that is generalized transduction, where a virus brings DNA from that cell to this cell, or that cell, same thing. Okay? And this cell can express a new gene For example, let's say it's this gene here, and it codes for tetracycline resistance. And this cell is sensitive to tetracycline. And then we bring in the tetracycline resistance gene. This happens in bacteria about 1 in 10 to the 5th, or 10 times more common than spontaneous mutation can make the tetracycline resistance gene. It can be brought into the cell by generalized transduction. Okay, any questions about transduction? Does transduction happen in human cells? Does transduction happen in human cells?
Yes.
I think somebody said yes. That is correct. In the cells of your nose, transduction is probably happening all the time. Okay? But in your germline cells, transduction is very unlikely to happen. And who cares if our nose cell gets DNA from another person by a virus? Because that nose cell will not go on to form an offspring. So transduction probably happens in the cells of our nose all the time. But in our germ cells, transduction is unlikely to happen. All right, any questions about genetic transfer?
No.
If not, let's go ahead and end this, and I will see you in the lab at 6:30 if you have questions. If you don't have questions, you do not need to show up. All right?
Thank you.
All right. I'll see you.